everyone, welcome to Vedantu NCERT Solutions 11th and 12th. I am Anshana, your biology master teacher, and today we are going to discuss question from the chapter called Cell Cycle and Cell Division. First question, what is the average cell cycle span of a mammalian cell? Okay, so we need to tell what is the cell cycle span of mammals. So in our textbook, NCERT textbook, it is clearly mentioned that human cell cycle span is 24 hours, right? 24 hours. So human is a mammal, so we can easily tell that 24 hours is required for a mammals to undergo cell cycle. Okay, yes. So how about yeast? Yeast is taking only 90 minutes. Okay, 90 minutes. How about E. coli bacteria? E. coli bacteria require only 20 minutes. Okay, so even though the human cell cycle happens in 24 hours, okay, it takes 24 hours, 23 hours in 24 hours, 23 hours for preparation. That preparation is known as interphase. Okay, interphase. So remaining 1 hour is for M phase. What do you mean by M phase? So in M phase, actual cell division. Okay, actual cell division happens in 1 hour. So that is known as M phase. Distinguish cytokinesis from karyokinesis. We need to define what is cytokinesis and also karyokinesis. Okay, so first let's look into cytokinesis. What do you mean by cytokinesis? Cyto means cytoplasm, right? Cyto means cytoplasm. Kinesis means what? Kinesis means to cut. Kinesis means to cut. So cytokinesis means cytoplasmic division. So during M phase, the cytoplasm is going to divide. So that is known as cytokinesis. Division of the cytoplasm. Division of the cytoplasm in the M phase. So that is known as cytokinesis. What do you mean by karyokinesis? Karyokinesis means what? Karyo means nucleus. Karyo means what? Nucleus. Kinesis means to cut. So division of the nucleus. Division of the nucleus in the M phase is known as karyokinesis. Describe the event taking place during interphase. Okay, we need to tell the what are the event takes place in the interphase. We know interphase is a preparation or preparatory phase. So in interphase, mainly 3 sub stages is going to happen. Okay, first one is known as G1 phase. So G1 phase is also known as gap 1 phase. What happens here? The cytoplasmic content will grow. Okay, the cytoplasmic content will grow. So cytoplasmic growth happens. Okay, cytoplasmic growth happens. What else? So RNA, proteins, okay, RNA, proteins and enzymes required for S phase is prepared in G1 phase. Okay, so next one, S phase. Okay, second phase is known as S phase. What happens in S phase? S phase is also known as synthetic phase. In S phase, synthesis of DNA happens. That means DNA replication. Okay, DNA replication happens here in the nucleus. And also, one more thing happens. In the cytoplasm, centriole duplicate. What happens? Centriole duplicate. Okay, so next one is known as, third phase is known as G2 phase or gap 2 phase. What happens in gap 2 phase? So the cytoplasmic content will increases. Along with that, what are the contents? That means RNA, protein and enzymes. Okay, RNA, protein and enzyme required for M phase is prepared here. What is G0 or quotient phase of cell cycle? Okay, so we know during cell cycle one cell is going to divide and produces two daughter cells. Okay, so these cells may undergo cell cycle again, or these cells undergo differentiation. These cells, so maybe one cell is going to divide and produces two cells, right? In these two cells, one cell undergoes cell division again and again, but the remaining one cell is here, no? So these cells undergo differentiation. So that means these cells metabolically active but do not proliferate. Okay, they are metabolically active but do not proliferate. So this stage of cell is known as quotient phase. So differentiation means what? The cell is undergoing some the cell is taking their function. So the meristematic cell is becoming xylem, phloem, parenchyma, collenchyma or sclerenchyma. Okay, so these kind of modification is happening in the cell, so that is known as differentiation. So the G naught cells undergo differentiation and produces different kinds of cells. Why mitosis is called equational division? Why it is called equational division? What happens in the mitosis? What happens in the mitosis? See, in mitosis, okay, so let's consider a cell. So this is the parent cell. This parent cell is consisting of four chromosome. Okay, so this cell is undergoing mitosis. So at the end of the mitosis, we will get two cells which containing 4 chromosomes each. That means there is no change in the chromosome number. There is no change in the chromosome number. So that is known as equational division. So this happens only in mitosis. How about meiosis? So think about meiosis. So in meiosis, what happens? The parent cell is containing 4 chromosome. But in daughter cell, what happens? The chromosome number become half, right? The chromosome number become half or reduced. So that is known as reductional division. In mitosis, we can see equational division. That means there is no change in the chromosome number. Name the stage of the cell cycle at which one of the following event occur. Okay, first one will say chromosome are moved to spindle equator. So that means, see, this is the cell. The chromosome are at the equator. That means at the center. So this we can see in metaphase, right? So metaphase. So second one, centromere split and chromatid separate. That means in the next stage, what happens? The spindle fiber will come and attach to the centromere, and this will pull from each side, right? The spindle fiber pull the chromosome from each side, so that the centromere split and chromatid move to opposite pole. So now we can see the chromatid moving to the opposite pole. So this we can see in which stage? That is called anaphase. That is known as anaphase. So next one, pairing between homologous chromosome takes place. So pairing. So that means we are talking about meiosis. So in meiosis, prophase 1 and prophase 2, prophase 1 of the meiosis 1 is very, very, very important, right? So what happens here? So 5 substages is going to happen in prophase 1. So first one is known as leptotin. So second one is known as zygotin. In zygotin, what happens? The condensed chromosome, homologous chromosome pairing happen, right? So chromosome number 1 from the parent, that is father, and chromosome number 1 from the parent 2, that is mother, come closer. So that is known as synapsis. So that happens. So that happens in the zygotin stage. Okay, next one. Crossing over between homologous chromosome chromosome takes place? Crossing over. Crossing over means recombination. Recombination means the gene between the homologous chromosome, non-sister chromatid of the homologous chromosome exchange happens. Okay, the genes of the non-sister chromatid of the homologous chromosome change happens. Exchange happens. So that is known as crossing over or recombination. That happens in the packaging stage. Packaging stage. Clear? Describe the following: synapses, bivalent, chiasmata. Draw a diagram to illustrate your answer. Okay, we need to tell what is synapses, what is bivalent and chiasmata. Okay, so first let's look into the synapses. What is synapses? Already we discussed, right? So in prophase 1 there are 5 substages is going to happen, in which second stage is known as zygote, right? So in zygote what happens? The homologous chromosome come close to each other. The homologous chromosome come close to each other. So that is known as synapses. So what happens? See, like this, the homologous chromosome come close to each other. So this is known as synapses. So next one is bivalent. What do you mean by bivalent? See, once the homologous chromosome come close to each other, what happens? So two chromosomes are very close to each other. So that stage is known as, that type of arrangement is known as, that synapses is known as bivalent. Clear? Yes? So next one is chiasmata. What do you mean by chiasmata? See, once the synapses done, that means two homologous chromosomes come close to each other. After that, this sister chromatid will split. That means these are the two homologous chromosomes which came together, and after reaching each other, okay, after reaching too close to each other, what happens? The sister chromatid will split. Okay, the sister chromatid will split. So after splitting, so what happens? The sister chromatid will split. After splitting, what happens? So one X-shaped structure happens. Okay, so like this, I'll show you. See here. See, like this. So the chromosome cross to that non-sister chromatid will cross each other. So this is known as chiasmata formation. How does cytokinesis in plant cell differ from that of the animal cell? Okay, let's see what happens in the plant cell and what happens in the animal cell. So you know what you mean by cytokinesis, right? Cytoplasmic division. The cytoplasmic division is known as cytokinesis. So in plant cell what happens? See, in plant cell, this is the plasma membrane. Outer to the plasma membrane there is one more membrane. So this is known as cell wall. Cell wall is very rigid. Okay, cell wall is very rigid. There is no means of furrow forming. So that's why what happens? So at the center of the cell there is a cell plate is forming. Okay, so which is made up of calcium carbonate. Okay, calcium pectate. Okay, calcium pectate. So this cell plate move to opposite means reaches the cell wall. Okay, this cell plate move to the cell wall and dividing the cell into two. Okay, so this is known as cytokinesis in plant cell. But in animal cell it is not like this. Means animal cell is having only plasma membrane. So plasma membrane is quite flexible. So at the beginning a small furrow is forming. Okay, so this furrow is deepens and reach at the each other. These two furrow reach at the middle and this forms two cells. Okay, this forms two cells. So this is known as cytokinesis in animal cell. Find examples where 4 daughter cells form meiosis are equal in size, where and where they are found in unequal in size. Okay, so let's see. 4 daughter cell we can see in meiosis, right? So in meiosis we can see 4 daughter cells are forming. Okay, so in meiosis, where this meiosis is going to happen? Only in gonads, right? Only in gonads the meiosis is going to happen. Clear? Yes. So in gonads, what is happening? In gonads, 2 main event is going to happen. In male, sperm production is going to happen, and in female, egg, right? So we know in human, lots of sperm is going to ejaculate, right? So in one ejaculation, millions of cells is going to discharge, right? So for that, each meiosis is going to produce 4 equal sized healthy sperms. Okay, but in females, in each month, only one fertile egg is going to develop. Okay, that means unequal division happens during egg production. In sperm production, equal sized cells are going to form. Distinguish anaphase of mitosis from anaphase 1 of meiosis. Okay, so anaphase of mitosis. What happens in anaphase of mitosis? See, this is the stage called metaphase. So in metaphase, what happens? The spindle fiber is attached to the centromere of the chromosome, right? Yes? Okay, so let's take 4 chromosomes. So 2 pairs of homologous chromosomes. So 4 chromosomes. Yes. So in what it is? In anaphase, okay, in anaphase of mitosis, for each chromosome, from either side of the spindle fiber is come and attach. Okay, so for each chromosome, from each centriole, there will be a spindle fiber is attaching. So when these two attached and pull, okay, so when these 2 spindle fiber pulling each other, what happens? The centromere will divide and what happens? The chromatids will split and move to opposite pole. Clear, right? Yes. So this happens in case of anaphase of mitosis. Let's discuss about anaphase of meiosis 1. Okay, anaphase of meiosis 1. What happens here? In anaphase of meiosis 1, there are 4 chromosomes. We'll take 4 chromosomes. Yes. So here the spindle fiber is attached to the chromosome, one of the chromosome of homologous chromosome. See, homologous chromosome means 2 chromosomes, right? Homologous chromosome means 2 chromosomes, in which only one is attached to one side of the spindle fiber. So that's why while pulling, whole chromosome is moving on one side. So here the homologous chromosome is splitting, but in anaphase of the mitosis, the chromatid is going to splitting. List the main difference between mitosis and meiosis. Okay, let's discuss the differences between mitosis and meiosis. Okay, so first thing is the mitosis happen in the somatic cell. Okay, mitosis happens in the somatic cells, but meiosis happens in gonads. That means testicles or ovary. In the testicles or ovary, the meiosis is going to happen and produces sperm or egg. In case of male, it going to produce sperm. In case of female, it is going to produce egg. Okay, next one. In mitosis, there is no change in the chromosome number. No change in chromosome number. But in meiosis, what happens? Reductional division. That means the number of the chromosome in the daughter cell is half. If at all, the parent cell is containing 4 chromosomes, the daughter cell is containing only 2 chromosomes. There are 4 cells is going to form. Okay, so in mitosis, what happens? There is no change. That means 4 chromosome in the parent cell. At the end of the meiosis, we will get 2 cells which containing 4 chromosome each. Okay, yes. So this is the main difference between mitosis and meiosis. What is the significance of meiosis? The main thing we can see that there is a scope for sexual reproduction, right? So here in most advanced organisms, we can see sexual reproduction. The sexual reproduction happens mainly due to the meiosis. If at all there is no meiosis, means the sexual after that there is no meiosis, means there is no reduction in the chromosome number. There is no reduction in the chromosome number, means the egg also containing 46 chromosome, the sperm also containing 46 chromosome. At the end of the sexual reproduction, the organism which containing 92 chromosome. So human means there is a certain exact number of chromosome, right? So that is 46. If it is becoming 92, means that is going to happen? Never, right? Even though it happens, that become humans. No, right? Okay, so meiosis helps in sexual reproduction. Second one is variation. It creates variation. You are known with, you are clearly seen what you mean by packaging stage, what is happening during crossing over, what you mean by recombination, everything, right? So that means there is an exchange between the genetic material. So this leads to variation. Variation leads to evolution. Evolution means a gradual change in the genetic material. So that leads to changes in the organism. So monkey to man you have come across, right? So that is evolution. So the evolution happens very slowly. Okay, very slowly. So monkey to man is not happen just in a day or just in a year, right? It took several thousand years. So that is known as evolution. So variation leads to evolution. So this is the significance of meiosis. Discuss with your teacher about haploid insect and lower plant where cell division occurs, and some haploid cell in the higher plants where cell division does not occur. Okay, so let's see. Haploid insects like drones, okay, drones of honeybee, and lower plants like algae, bryophyta, where haploid cell is going to produce. Haploid cell is going to produce, or haploid cell is going to divide and produces new organism. Okay, so haploid cell undergo mitosis and produces new organism. Okay, so next one, some haploid cell in higher plants where cell division does not occur. So like egg and sperm, the egg and sperm does not undergo cell division. So once it becoming egg, or once it becoming sperm, there is no cell division occur. So these two cell is going to fuse and produce a zygote. So this is the second case. Can there be mitosis without a DNA replication in S phase? Without a DNA replication, we can see mitosis? No. No mitosis without DNA replication. So first the cell undergo DNA replication, then only mitosis is going to happen. So you know what you mean by mitosis. In mitosis what happens? So the daughter chromosome, the daughter cells containing chromosome is equal to the parent cell. So that means parent cell containing 4 chromosome means the daughter cells also containing 4 chromosome each. Okay, so that require DNA replication. Can there be DNA replication without cell division? Yes, we can see. Yes. How we can see this? See, cell division happens before DNA replication. Cell division happens after DNA replication. That means first DNA replication is going to form and the content of the DNA become doubled. That means 2C numbered DNA become 4C. Only after 4C the cell is going to divide. Analyze the event during every stage of cell cycle and notice how the following two parameters change. Number of chromosome n per cell, amount of DNA content C per cell. Okay. So first let's see. See, this is the cell cycle. In cell cycle, what happens? Interphase. So this is G1 phase, this is S phase, and this is G2 phase, and the remaining is M phase. In M phase, prophase, metaphase, anaphase, and telophase. Okay. Finally, cytokinesis happens. Okay. So here in G1 phase, what happens? In G1 phase, the cell is preparing, right? So in G1 phase, the cytoplasmic content will increases. So the growth happens, right? So here the chromosome number is 2C and 2N. Okay, 2N, and DNA content is 2C. So at the end of the G1 phase, the number is same. There is no change. Okay. In S phase, what happened? DNA replication happened. Okay, DNA replication happened. So what happens here? Yes. So what happens here? At the end of the S phase, the cell which containing chromosome number, there is no change in the chromosome number 2N itself, but the DNA will increases. That become 4C. Okay. So G2 phase, what happens in the G2 phase? The cell is preparing for M phase. There is no change in the G2 phase. The chromosome number or the DNA content will not change. Okay, the same. Next, in prophase also same thing. Nothing happens. Okay, so nothing happens. Metaphase also nothing happens. The chromosome come and align at the equatorial plane. So 2 and 4C. But in anaphase, at the end of the anaphase, what happens? A cell which containing here 2C, here 2C. Okay, so here 2N, here 2N. Okay, the chromosome number becomes same. Means there is no change in the chromosome number, but the DNA will segregate. Okay, at the end of the telophase, there will be 2 cell which containing 2C and 2N in number. So DNA content reduced, but the chromosome number, there is no change in the chromosome number. Clear, right? So this is about the questions from the cell cycle and cell division. Hope you have understood each and every questions. So if as, like our video, share it to your friends and subscribe. Thank you all.